Hello, everyone. My name is Kenny Coogan. I'm the Education Director for the International Carnivorous Plant Society, and welcome to another exciting monthly webinar. Sometimes we have more than one a month. Today, we are joined by David Armitage. He's going to do a presentation about Darlingtonia. If you have any questions for him and you're attending this live, you can type them into the chat box at the bottom of your screen, and I will read them to him at the end. And with that, David, thank you for agreeing to this, and it's good seeing you again.
Thanks. Yeah, it's great seeing you too, Kenny. So thank you all for attending this talk. I'm kind of sorry about the time if it's inconvenience. This is 9 a.m. Japan time, so this is like literally the earliest I can function in the morning without sounding crazy. I might still sound a little crazy though. Today I'm going to talk about some of the research that I've done and I'm currently doing with my lab group on this beautiful pitcher plant here, Darlingtonia californica. So I'm going to focus mostly on studies concerning the natural history of Darlingtonia. A lot of my work also uses this plant sort of as a natural microcosm to study processes like microbial community assembly and very basic ecological questions. But today I'm going to focus mostly just on the natural history and how and some of the things we've been doing there. So just a background quickly on Darlingtonia. It's in the family Sarraceniaceae. You can see this beautiful phylogenetic tree here with Darlingtonia highlighted down here. You can see that it's kind of an outgroup to the rest of the Sarraceniaceae, so Helianthera down here. And Sarracenia, so the North American pitchers up here. So one thing that might strike you already is the fact that Darlingtonia is also a North American pitcher plant, but it's actually, sort of an outgroup, and Helianthera and Sarracenia are more closely related to one another, despite their, large geographic separation. Here's a pressing of Darlingtonia that was made about 20 years ago now, I think. And you can see the plant itself bears a lot of the normal Sarracenia-like morphology, but with some notable exceptions, mostly being the shape of the distal end of the pitcher leaf. It's kind of curved over in a manner similar to like Sarracenia psittacina, for instance. The flowers are also quite different than the rest of Sarracenia. And generally the flowers are produced in the same way, or the leaves are produced in the same way in sort of a whorl-like structure with between like 3 and 8 or 9 new leaves being produced each year. And then the year's largest leaf is usually the first one to come out. And the leaves last anywhere between 1 and 2 years, depending on weather conditions and things like that. So here's a closer look, of an insect's eye view maybe of Darlingtonia, and you can see these sort of clear, pigmentless fenestrae in the top of the pitcher leaf, which are a putative mechanism for tricking insects into thinking that they can just fly directly up and out of this this leaf cavity here. But they soon find out that they can't, and they are then trapped and funneled down into the trapping chamber and digestion area, which is at the the base of the leaf. And you can see this is a particularly satiated pitcher leaf here with hundreds, if not thousands, of what look to be beetles. There's also some yellow jackets in there. They mostly seem to eat beetles and yellow jackets from my observations. And you can also see this sort of interesting gradient of decomposition. So at the base of the leaf here, it's essentially feces at this point. And at the, on the right side of the leaf, you got the undigested prey still. And unlike a lot of the Nepenthes, Darlingtonia really don't produce really strong digestive fluids. Their fluid in these leaves is relatively neutral, like between pH of 5 and sometimes up to 8. And so bacteria actually do a lot of the digestion via the enzymes they produce. So it's sort of a mutualistic interaction between the plant and the bacteria that they feed. Again, like a digestive system. So that digestive system doesn't just comprise bacteria. We have a number of other what are called inquiline species that live within the pitcher chamber, most notably these mites here, Saraceniopus darlingtoniae, and these midges, the larvae of which you can see here crawl all over and chew up the remains of the insects. And through the actions of just shredding up the insects, defecating, making the surface area available to bacteria to finish the digestion, what ends up is all of the nitrogen and phosphorus that start as organic compounds on these dead insects end up as essentially liquid fertilizers. So nitrate and ammonium that then the plant sucks up and uses to feed itself. Essentially a root above the ground. And there's also a number of other things like protists and rotifers and whatnot, which I'll show you in a sec. So here's a microscopic image of what's going on inside of a typical Darlingtonia leaf. You've got your Metrionemus midge larvae here. You got a few nematodes swimming around. And I think the camera will then zoom down, hopefully, and you'll see a pasture of these mites. Yeah, here we go. Grazing like cattle, basically, on the digested remains of these insects. So these are all the Saraceniopus mites. And it's thought that the mites and the midges are obligate inquilines in these in Darlingtonia. So nobody's found this species of midge or mite anywhere outside of these pitcher plants. So there is potentially some very intricately co-evolved relationship there between the two. Zooming in a little closer, we've got ciliates. So these are, like paramecium kind of things, swimming around and consuming bacteria and detritus. And then if you look very closely, you can see thousands and millions of little bacterial cells swimming around as well. And a fun fact, when I was actually studying Darlingtonia and the bacteria living inside of the leaves, we found that the density of microbial life inside of like a milliliter of Darlingtonia pitcher fluid at the height of the growing season had densities of bacteria that were higher than almost any other substrate on Earth. And essentially, the only other substrates that had bacterial loads as high as these pitcher plants would be like kimchi bioreactors, so like fermentation reactors in industry, and human feces. So quite a microbially rich soup that you can immediately tell it's full of decaying microorganisms when you look at it and smell it. Zooming back out to the whole range of the species, you can find them growing everywhere from this little area in Sand Lake, Oregon, just sort of southwest of Portland here on the coast, all the way down throughout coastal Oregon. At which point, when we get down to the Siskiyous, they kind of go inland a bit, and we have a very large cluster of large populations of Darlingtonia in Northern California, Southern Oregon regions, as well as a few other populations in the Trinity Alps here in California. And then in the Northern Sierra Nevada mountains are a few discrete populations. And so if you look at these things, they, like many other species of pitcher plants, display an extremely highly fragmented distribution. So, zooming in, you can see even at the very local scale, you've got like all of these separate patches here corresponding to locations, locations with just the right conditions for growth. And so these are some of the most fastidious species that I've ever encountered with regards to their habitat requirements, and they often require, they tend to require flowing water moving over their roots that can't get above a certain temperature. And there's some interesting biochemical reasons for why that might be. But yeah, you tend to find them in these kind of unique little montane seeps with available cold water. Another thing you want to look out for when you're looking for Darlingtonia and trying to associate it with some landscape level feature are serpentine soils. And so serpentine is an ultramafic soil that is distributed, comes from the seafloor. It's generally very low in nitrogen and high in metals like magnesium, cobalt, and nickel and iron. And you can see that the outcrops, some of the major outcrops of serpentine soils corresponds quite closely to the distribution of Darlingtonia, at least where it exists in the mountains. Along the coast, you do find some serpentine soils where you find Darlingtonia, but generally along the Oregon coast is quite different with regard to the soil types and the habitats in which they live. Finally, precipitation also seems to be a strong determinant in their distribution. So you can see, areas with higher precipitation in the mountains of California, and as well as coastal Oregon correspond to where they're distributed as well. So in 2022, when I first started my faculty position here in Okinawa in Japan, I immediately got some money to then fly back to the United States and complete this sort of life goal trip of mine, which was to basically procedurally drive from Portland and hit every single known Darlingtonia population throughout its range, collecting both specimens of the plant itself, as well as specimens of the mites and midges that live inside of the leaves, to study all sorts of things about their genetic structure across the landscape. So, identifying perhaps where the genetic diversity in these populations is highest, how much interbreeding might occur between these populations, things like that. And so you can see here my travel partners are my wife Katie and my son Cyrus. We're up in looks like Gold Beach, Oregon, just hiking up the side of a mountain looking for plants. And then we've got my graduate student who is currently writing her dissertation on this work, Nono Hasegawa, pictured here, as well as Asa Conover, a graduate student from UC Berkeley, who is also a Darlingtonia enthusiast. And so they all joined us for this trip as well. So right now, I'm just going to walk you through some of the varied habitats in which you can encounter Darlingtonia, with just keeping an eye on how different they all actually are from one another. So here's the northernmost population. This is in a place called Sand Lake, Oregon. It's right on a roadside, which is kind of funny. But if you can't really see the plants here, but this is the habitat it grows in, very shaded, which is somewhat atypical for Darlingtonia. Often when you're looking for Darlingtonia, you're not looking for like ferns and like equisetum and things like that. So the flora here is quite different than what you would typically find Darlingtonia growing in. Zooming in a little bit more, maybe you can see some pictures start to emerge there. But generally quite hidden, a very small population, and the individual plants and leaves are quite diminutive as well, perhaps reflecting this very northern distribution. But as you can see, I mean, there are leaves being produced here, regularly every year, as well as flowers. Moving south, this is a very famous site at the Darlingtonia Wayside in Florence, Oregon. If you've ever gone on a Darlingtonia pilgrimage, as I have, you've probably stopped here before. It's a great site, really large population of Darlingtonia, great boardwalk, so really accessible. And there's some debate as to whether or not this is truly the northernmost natural range extension of Darlingtonia, because the status of the Sand Lake sites and other sites north of here are still a little bit contentious. Were they planted? Perhaps, we don't know. Here's another site at Lake Wannock, just south of Florence. Quite a different site than what you found just nearby. This is a lake, and this is there's a lot of little moving, meandering streams moving amongst large mats of sphagnum moss. So this is the kind of site that you typically, like if you're from the east coast of the US, would be finding a lot of Sarracenia purpurea growing in. Kind of a classic purpurea looking site. But here on the west coast, we've got Darlingtonia growing. And here's a close-up of what that looks like. And again, there's not a ton of sites that have this type of characteristic. So quite unique, I think. Here's another site down further south. Basically, over this sand dune here is the ocean. So I believe this is probably the site, one of the sites closest to the ocean at which Darlingtonia grows. And again, quite diminutive plants, very small population, but they are growing here in this kind of weird exposed brackish saline condition. But there is some spring water that's flowing across this hillside that's kind of keeping them persisting. Another site in Oregon, just nearby that previous one I showed you. Again, we've got a large floating mat of sphagnum moss. And as I tried to climb across this log, I actually fell in the water here. So, this site holds a lot of a lot of negative energy for me. But you can see the pictures right there. Again, a small population growing on one of these floating sphagnum mats in relatively deep water. Another site's very close to the coast. This is Cape Blanco, I think, in Oregon. And this is like just a grassy savanna. But here, again, some Darlingtonia growing, again, very close to the coastline. And finally, once we get down into the Siskiyou Mountains, you finally start seeing some of these, serpentine outcrops and Darlingtonia growing in, I guess, what's kind of considered their most classic habitat, these spring-fed rocky seeps here. So this is from the western side of the Siskiyou Range, or in the sort of wetter side, just north of Crescent City. Here as we drove up to this site, this is an example of what you would regularly see on some of these steep, winding road cuts. You'd find Darlingtonia just kind of growing amongst the cracks wherever there's water flowing down. And these areas are just like, they have patches of Darlingtonia where it's nothing but this plant. So probably the region of the US that has the highest, or the region of their range, I should say, that has the highest density of these plants. On the eastern side of the Siskiyous, it becomes a little bit drier, but nonetheless, here's where you find some of the largest populations of Darlingtonia growing. So this is the region around Quincy, Oregon, $5 or $8 Mountain, great places to visit. And even in places that you wouldn't normally expect them to be growing in because you would think it was too dry, you can actually see these plants have achieved the ability to colonize and grow a little bit. Even in some of this looks like just the remains of a gold mine, for instance, and so this population might be relatively young. Here, then, we've moved down to the Trinity Alps in sort of northern central California. And here we find the highest elevation populations of Darlingtonia, specifically on Mount Eddy in this area called the Deadfall Basin. And I think maybe there's some higher populations, but I can't think of any right now. I think right around here maybe is the highest, just south, just down from that lake there. The plants here are, again, quite short, diminutive looking, but huge populations. I mean, you can basically just follow a continuous spring-fed seep all the way down the mountain, and it's like nothing but these plants growing for kilometers. But yeah, generally quite small relative to the massive leaves that you would find up in the Siskiyou area. Then finally, we go down to the site that I've primarily been working at in Quincy, California, in Plumas County. And I was really worried about visiting this place because the year prior, from July to October, there was what I think at the time was the largest forest fire that had ever been observed in California. It might have been, superseded in the past few years. But 390,000 hectares burned, which is surprisingly 3 times the size of the island that I currently live on. So troubling, and we were very worried because when we got to this site, this is what the forest looked like, just totally charred. So we went back to the places that we knew Darlingtonia were growing, and we were actually very happy to find that all of these spring-fed seeps were relatively untouched by these fires. And in fact, a lot of the wood had fallen, kind of blocking off and allowing for a bit more water to kind of pool up and be built up there. So potentially even improving the habitat. And so here's an example you can see this charred forest. This is sort of where the fire line went, and then everything else was not touched. Here's a really famous site called the Butterfly Valley Botanical Area right outside of Quincy, California, in Plumas National Forest. This photo here was taken in 2014. This is where I did most of my research back when I was a graduate student at Berkeley. I returned to this exact area in 2022, and this is what it looked like. Very shocking, right? I was extremely depressed to find this entire patch of plants just dead. And the reason why I believe was because the traffic there's a road basically just down from this site. And I think just like car traffic and maybe people even visiting this site had compressed the dirt on this road to such an extent that water, spring water could no longer permeate through the soil there, thereby drying this area out. Luckily, though, I think 2 or 3 years ago, when this die-off was first kind of being observed, this road was like immediately closed off. So now you can't drive down this road anymore. And there was actually a boardwalk installed. So people like me aren't just trampling through this fen, and you kind of have to stay on the boardwalk. So, and if you actually look at these sites very closely, you'll see some regrowth occurring as well. So promising conservation effort here. And it's a great job. I think the Forest Service was responsible, and possibly the California Native Plant Society as well. Some of the southernmost sites here are, again, at slightly higher elevations. This is a place called Harris Meadow, which is fiendishly difficult to access. This was kind of the sketchiest place that we drove in our rental car. But again, these are very different looking habitats. I think these are actually quite grazed by, I don't know what kind of animal, farm animals. But here you can see just another large population of Darlingtonia growing in this beautiful natural setting. And here finally we have the southernmost population of Darlingtonia growing in a place called Grouse Ridge. This is private land that the owners were gracious enough to allow us to come sample. And here, I believe this is the site where the anthocyanin deficient Othello strain of Darlingtonia was discovered years ago. So we didn't actually see any when we were looking, but so again, documenting our trip, we're doing the pitcher plant stoop here, looking around, collecting plants, collecting midges and mites, sucking them out. And in the lab then, here's Nono doing some lab work, extracting DNA from these plants. We then sequence the DNA that we extracted from both the plants, the mites, and the midges to learn something about their population structure. So a few findings. First, we visited 3 sites that are putatively, maybe introduced. So there's this famous site down near Mendocino in this forest called Albion Bog, where you find Darlingtonia. You find a few mites in these pitchers, but absolutely no midges. as well as in this site here, this campground called Cherry Hill, where you again find no mites and no midges in these pictures. And then up here in Sand Lake, you find, I think we found one individual midge and again, no mite symbionts. Contrasting that with all of the rest of these sites, at like, almost 100% of these sites, we were finding both mites and midges within all of the pictures. So perhaps there's some sort of dispersal limitation preventing the midges or mites from locating these potentially introduced populations here. The population structure of these plants can kind of be summarized by this figure here, where each color represents sort of a statistically estimated population level grouping. So wherever the color is the same, that means that the site here, which is on the y-axis, would be considered more of a genetically homogeneous population that breeds with itself rather than from these different populations. So what we found here was first that there's somewhere between 4 and 5 distinct genetic clusters, or we can call them unique genetic populations of Darlingtonia, that sort of follow this latitudinal gradient here, where the Oregon populations, we've got the Trinity Alps population and the Sierra population. The little population down in this Albion bog is clearly derived from somewhere up here in the Sierra Nevada mountains, which is kind of an interesting finding, I think. And then there's sort of maybe a couple, maybe 2, or maybe just a single population, but perhaps more gene flow occurring just within this region. So you can see that here where you have perhaps a little bit more genetic admixture between these 2 populations than with the rest of these sites. We can also then look at genetic diversity. So within an individual patch of Darlingtonia, if we start randomly sampling the genetics of each pitcher plant, how much genetic variation is contained within each one of these populations. And so that's what I'm showing here in this graph, where on the y-axis we've got within sites genetic diversity, and then here I've sort of sorted these sites out by their distance from the center of Darlingtonia's range. So the star here represents sort of the geographic centroid of where these plants are, and we find that there tends to be a pretty significant sharp decline in within population genetic diversity as you move away from this population center here. So generally the least diverse populations are those at the margins. We can graph this out too and make a little map of it. Again, so this is a graph of genetic diversity of Darlingtonia across space, where the highest genetic diversity is found in the Siskiyou, Northern California, Southern Oregon mountains, with declines going south and north. We can also ask which of these populations is most genetically distinct from the average. And so to do that, we basically just ask, we find sort of a very, average genetic point here, this triangle, and then measure each population's genetic divergence from this average. And so points that are sort of farther out from this triangle represent those that the genetics tell us are more unique compared to everything else. And generally what we find is that some of these marginal populations, so this, again, Grouse Ridge population up here is, the farthest south population. A lot of these Oregon populations and small populations also tend to be more genetically distinct, possibly through the joint effects of genetic drift and also just isolation from migration from a lot of, other populations that might surround it. Here now is a phylogenetic tree, so a tree representing the relatedness of all of our individual Darlingtonia populations, linked to the same genetic tree showing the relatedness between the midges that we recovered from each population. And so what this suggests, this is a very preliminary analysis. I made this graph yesterday, but it seems to indicate that the midge symbionts show somewhat of a similar population genetic structure to the plants themselves. Although there are some noteworthy exceptions, like here we have, the Oregon population and some of the populations in Trinity Alps seem to be more closely related for the midges, but for the plants themselves, that isn't the case. So perhaps there's some topological differences here, but there is still, I think, significant population clustering. So the midge population can be somewhat predictive of the plant population. Now I'm going to move a little bit into some work that I've previously done. Here you can see some ants that I've thrown into some pitcher fluid or into some water. And because I spent so much time out in the field studying these things, I got bored and was just, spending my days drowning ants in pitcher fluids and watching what they do. And I quickly noticed that these ants actually drown much quicker in the pitcher fluid than they do in sterile water. And you can see that the ants in the water haven't even sunk to the bottom of this yet. Whereas for all of the Darlingtonia pitcher fluid or the enrichments of bacteria from these pitcher fluids, they're just dead, stunned at least, compared to the ants in the water. So perhaps there's something going on in the fluid properties that are imparted either by the pitcher itself or by the bacteria living in the pitcher that might aid the plant in capturing prey. So that was the study that I wanted to do. And thankfully, I had some friends who knew more about physics than I did, and they showed me how to take a photograph of a droplet of pitcher fluid and use that to then parameterize this equation down here, which gives you a measurement of the surface tension of that fluid. So again, the idea here is that we can ask, well, how does the surface tension of pitcher plant fluid differ from that of water or bacterial cultures from bacteria derived from these pitcher leaves? So we did this. We digested, wasps with pitcher fluid and made a bunch of, bacterial enrichments. And then you can see the some of the results here where we have now interfascial. So this is surface tension. This is water, the first thing here on this graph. The rest of these are either natural Darlingtonia fluids, which are significantly lower in surface tension than water. And then the enrichments of the bacteria, so growing the pitcher plant bacteria in sterile water, then lowers that surface tension as well. So what are the functional consequences of this lowered surface tension? Well, again, I was just throwing a bunch of ants into pitcher fluid and monitoring how often they were able to escape or not. And so here we have four different groups of pitcher fluid. We've got the bacterial enrichments, and out of the 50 to 100 ants I threw into each one of these, 100% of them, or nearly 100% of them, were retained and were unable to escape the pitcher fluid. But actually, almost all of them were able to escape when it was just pure water. And so then I diluted this pitcher fluid down with pure water to parameterize this graph here, which basically shows as you dilute pitcher fluid with water, the probability that an ant thrown into this fluid will escape increases quite predictably, so that there does seem to be some effect of the actions of the bacteria in the pitcher plant that have nothing to do really with digesting the prey, but simply just helping the plant capture the prey and stun it as well. Another thing that I was always really interested in was this bizarre appendage on the pitcher. This is, why they call it a cobra lily, because it's got this sort of forked tongue apparatus. And if you read the famous literature on Darlingtonia, they always talk about how these wings, fishtail appendages, are used to attract prey. Here you can see, 3 different examples of people just writing in saying that, yeah, these things are adaptations by this plant to attract prey. And so I just wanted to test that. Like, how important is this tongue for successful prey capture on the plant? So we did a really simple experiment in like 2015, 2014, where we went out and just removed the tongues off of pitcher plants, either at the individual leaf level. So we would have a leaf from one plant with its tongue, and then a paired equally sized leaf from the same or a nearby plant without the tongue. Or we would just remove tongues from like a whole big patch of pitcher plants to then ask, well, does the removal of this tongue, when we go back and measure how much insects they've captured, does it affect it at all? And I guess kind of surprisingly, not really. So this paper basically posited that, under a natural condition where we have the tongue, here's the prey mass on average for each leaf. And then if we remove the tongue on this leaf, it doesn't really seem to statistically differ from one with its tongue appendage. And maybe it's a little bit lower if we remove all of the tongues from a single population, but again, it's still not statistically different numbers of insects that are captured here. And so that led me to kind of wildly speculate in this paper on the potential role of this tongue appendage. And one of the potential hypotheses is that it could be just like a facultative surface for expressing photosynthesis. So here we have pitcher plants, Darlingtonia, grown in either the shade or the sun. And I went out and just measured the chlorophyll contents in just the tongue appendages between these different populations, and found that in shaded habitats, there does seem to be quite a bit, significantly more chlorophyll found in these tongues than in the sun. And here's kind of what that looks like, right? You've got a very green plant growing in the shade versus, a reddish, kind of whitish tongue here in these sunny populations. Here's another study that we're currently doing out in California, where we started this a few years ago, and ASA is now maybe in the fields as we speak, capturing wasps and hanging wasp traps out in these pitcher fens. And the idea here is that we want to test this prediction that was made by Daniel Joel in this great paper from 1988, which basically speculated that we need to recast the relationship, perhaps, between insect prey and the pitcher plants, rather than being sort of a predator-prey antagonistic relationship. If you were to sort of average it out over each individual plant and insect within a population, it could be more of a mutualistic, mutually beneficial relationship. And the reason for that is because only a really very small fraction of wasps, say, that land on a pitcher to feed on its nectar end up getting trapped in it. I think Aaron Ellison and colleagues wrote a great paper where they sat around and filmed or just watched Darlingtonia for like a week or something straight, and found that they only observed a single wasp being trapped out of like 10,000 that visited these plants. So the idea here might be that maybe wasps or other insect prey items are benefiting as a population and feeding, through the nutrient provisioning that this plant provides them, and only a few are actually sacrificed. So we can study that by asking, does the fraction of nitrogen in wasp or potential prey species near pitcher plants reflect some large dietary contribution of those plants to the insect populations there, versus those same insects elsewhere that don't get to benefit from those from that freely available nectar. So to do that, we use these things called stable isotopes, which are in here we're using a stable isotope of nitrogen, where this isotope is naturally abundant in the atmosphere at a percentage of like 0.03% or something. And it differs from sort of the normal nitrogen, 14N, that we all know and love, simply by the addition of an extra neutron. And so it's slightly heavier than the most abundant form of nitrogen, the most abundant isotope. And through the actions of feeding, so like through the actions of fixing that nitrogen from the atmosphere and then moving it up through the food chain, digestion and metabolism sort of differentially favors, the heavier nitrogen or the lighter nitrogen in different proportions. And so what we can do is use the relative abundance of this 15N isotope to estimate what kinds of foods things are feeding on. It's sort of a marker of trophic level. And so here we can first go out into our field of Darlingtonia. This was done at Butterfly Valley Botanic Area, and we measure the non-carnivorous plant ratio of 15N relative to air. And you can see that it's variable, but it's quite low. We can then go out and measure the same thing on Darlingtonia and find that it's always going to be significantly higher than its non-carnivorous plants in the same habitat. So this reflects the fact that these things are eating meat, eating insect carcasses, rather than either fixing nitrogen from the atmosphere or using inorganic nitrogen. We then place these wasp traps directly over the pitcher fens and capture the wasps. And we either do this, right above the pitchers or somewhere else in the forest where there are no pitchers. So here's the nitrogen isotopes from the wasps that are not near the pitcher fens. And so what we want to know then is if wasps at the pitcher fens are significantly benefiting from feeding on something with approximately like the exact same 15N concentrations as non-pitcher fen wasps, we should hope to find a slight enrichment of 15N in the wasps from these pitcher fens as potential evidence that they are feeding on these plants and on the nectar that they produce. And there's the prediction that they'll be enriched in 15N. And so we went out, and these are the 15N signatures from the wasps that we collected. And although they look kind of similar, there is like a small statistical difference here. So, the pitcher plant associated wasps do have a slightly higher elevated 15N, reflecting, possible contribution from these plants, which are the most abundant plant around. We're now running this experiment across multiple other geographic regions to see if this pattern generally holds. The final thing I'm going to talk about, maybe more of a doom and gloom type of analysis. When I was out in the fields at certain sites, I noticed that there, especially at some of these higher elevation sites, quite a bit of mortality and sort of loss of groundwater flow that was, otherwise, not happening like even 10, 15 years ago when I last visited some of these sites. Here's a site, I think this is by Lake Eleanor in the Trinity Alps, where here you have this sort of big dead or dying population of Darlingtonia that's sort of dried up. And here is, half of the remaining population now. If you look, it's not like the one at Butterfly Valley where there's now regrowth. There doesn't seem to be any regrowth going on here. And so, this kind of got me a little bit alarmed because I was seeing this quite a bit at a lot of different sites, the drying up and mortality of these plants. In Oregon, a lot of the sites in Oregon also seem to be affected by this as well. And so I basically just, I'm working now on doing a lot of sort of statistical forecasting of how and where plants might be able to move under future climates. So the idea here is that we can take, our occurrences of any plant species, let's say this is Darlingtonia, and create statistical habitat suitability models using information about soil characteristics, climate characteristics, human population densities and things, to then create models that first show our, current suitable habitats for Darlingtonia, as well as then project these forward through climate, future climate scenarios, and ask, well, where is the distribution of our particular species projected to shift, shrink, grow, et cetera, maybe 100 years from now, given some, emissions model. So I did this using sort of a intermediate level of emissions model where I guess like it wasn't totally out of control, but wasn't really that in control either. So what I'm going to show you now are just the current habitat suitability model for Darlingtonia on the left-hand side here, and then the future projected habitat suitability model for Darlingtonia for the year 2100 on the right-hand side. And so areas that are the brightest red are the places where we would anticipate the climates and soil and other conditions to be the most amenable habitats for the plants themselves. And you can see that they are, generally quite reflective of the observed distribution of Darlingtonia from this 2024 model. And then if we project this into the future, you see first a lot less red on this map, which is, here not a good thing because it does suggest that the highest suitability habitats will shrink in size quite dramatically. And we're looking at potential largest losses down here in the Sierra Nevada mountains. So another thing to note is that there's not really like any northward movement of suitable habitat. So it's not like, as the climate changes and the northern hemisphere warms up, these plants can necessarily just migrate northwards. And the reason for that is because there's all of these other non-climatic factors that are also really important for determining their distribution. So serpentine soils, the availability of cold flowing spring water. These are things that are still constraining the distribution of Darlingtonia. And so, they're kind of like similar to maybe redwood trees in that they're sort of trapped and potentially unable to move anywhere in the next 100 years. And finally, I also created this simulation of habitat connectivity, so sort of population movement distances between these different sites. So the idea here is that these red lines kind of trace the shortest possible routes that either pollen or seeds from different populations can move between known sites of Darlingtonia. And what I did here was basically measure the overall average distances between sites sort of across these like least cost path differences. So you can see here, right, like the fastest way to get from this southern Sierra sites up to maybe the Trinity Alps is along this pathway here. I did the same thing under future environmental scenarios, so climate change scenarios, and then asked what is the average distance between populations, sort of the functional different distance between these populations, and found that on average, we predict that it's basically going to take a lot longer for genes, pollen, and seeds to be exchanged between these populations in the future than it currently is. And so this has some general ramifications for potential genetic isolation, right? If it takes longer and if it's becoming harder for two populations to remain connected via gene flow, it basically means that more isolated populations could accumulate, deleterious mutations, maybe less able to adapt to climate change than they otherwise might be if they have beneficial alleles and mutations moving into them with more ease. So hopefully maps like this can help us identify, corridors that we might need to prioritize for conservation, habitats, like maybe some of these southern Sierra or northern Sierra habitats that we might need to prioritize as well. And so with that, I'll just thank you all for listening, and thank my funding agencies, my current university, OIST, my previous university, Berkeley, and none of this work could have been done without especially the help of Oregon State Parks, U.S. Forest Service, U.S. Department of the Interior, and the Siskiyou Field Institute, which is it's in Quincy. It's by Quincy, Oregon, a beautiful place that does a lot of great outreach and field botanical courses and stuff. And so I encourage you all to visit if you ever get the chance. Thank you.
I'm Kenny Coogan, the Education Director for ICPS. And the ICPS is an organization of horticulturists, conservationists, scientists, and educators, all interested in sharing knowledge and news of carnivorous plants. We have been around since 1972. That's a long time. And we started with a carnivorous plant newsletter. And we're a group of lots of different people from all over the world. And by joining the ICPS, by being a paid member, you can help our society flourish. It has evolved into a truly international society. I met David in Japan, and 52 years ago, it was Japanese and Germans and the US. And now we have representation from all over the world with more than 1,400 paid members. We offer a lot of free webinars coming out. I've been the education director for a couple of years now, and just this past year, we've started having more than one webinar a month because so many people are interested in it. If you go to our Facebook page, you'll see the upcoming list. And this bottom link, icps.clubexpress.com, the M got cut off. But if you go there, that's our official website. And that also lists all of the upcoming webinars. If you're interested in being a presenter, you can email me at kenny@carnivorousplants.org. A couple of years ago, I created 5 animated videos. They're perfect to play for kids and people and adults who are just getting into the carnivorous plants hobby, passion. One video is about growing carnivorous plants inside. One video is about growing carnivorous plants outside. One is feeding Venus flytraps. One is about acclimating carnivorous plants. And one is, what is a carnivorous plant? Another initiative that I took on was a member suggested that there should be a World Carnivorous Plant Day. And 4 years ago, we made it happen. It is the first Wednesday of May every year. So this year, May 1st, 2025, that's the Wednesday. We wanted to do it in May because a lot of carnivorous plants, although they're from all over the world, a lot of them and a lot of the growers are in the northern hemisphere, and May is a good time when all the carnivorous plants are waking up. And we wanted to pick a day of the week so kids can be celebrating it inside the classroom. And as David knows, we had to avoid the first Saturday of May because that is World Naked Gardening Day. And we did not want to combine the 2 days. If you become a paid member, you can go to this website. The more years you buy, the steeper the discount. And the benefits of becoming a member, in addition to supporting conservation and education, is that you get access to our seed bank. You can email our seed bank manager. We can send seeds to many countries. Just contact Mark to learn more about that. And I think the real feature of being a ICPS member is that you get a quarterly colored newsletter. And this features regular columns about horticulture, research papers, field trip reports, conservation news, books and literature reviews, and then there's also communication for members. This is the official place to register a cultivar. A cultivar is a cultivated variety. So if you're breeding things and you see something that's really special, you can register it with the ICPS. We also post artwork, and we also do meeting announcements, and there's some advertisements in the Carnivorous Plant newsletter. So that's, I think, the real benefit of being a ICPS member. But once you become a paid member, you get access to the last 52 years of newsletters. They're all digitized, and you can see like, oh, wow. And this year, this plant was discovered, and now it's really common and things like that. Another initiative that I took on was Carnivores in the Classroom. This, I think, is the third year. We hold the grant period in the month of August. Teachers in K through 12 classrooms can apply. And then what they win is $150 of live carnivorous plants shipped to their classroom. And in order to win those plants, the teachers need to create, actually, I say teachers, but in the past, we've had several students submit lessons on behalf of their teachers, and then they win $150 worth of plants for their classroom. So students and teachers can submit lessons. We have 3 categories, K through 5, then we have the middle levels, and then we also have high school levels. And what's really cool is anyone, homeschool parents, teachers, everybody can go to our website, carnivorousplants.org, and you can see all of the lessons, and you can download them for free, and you can utilize them. And the sponsors of this year are listed here. These are nurseries and individuals who directly send live plants to those teachers. And in the middle, we have Carnivorous, which is a 3D printing company located in Canada. And they sent each of the 35 plus teachers a free 3D printed carnivorous plant. And if you or your child has a 3D printer, you can go to our website and the instructions are free and you can print your very own carnivorous plant. We have 5 designs, which are seen right here. If you want to support our education and conservation initiatives, in addition to being a member, you can go to our drop ship website. And this is where you can buy tote bags, t-shirts, mugs, things like that with different carnivorous plants on them. And all of the proceeds go to the education and conservation. Something that's special about the Carnivorous Plant newsletter is here's just a brief sample of the last quarter newsletter. We're a volunteer organization, but we're an increasing voice in the world of conservation. We would also like to excel in promoting awareness of nature and the interaction between people and plants, which David did a great job on. So by becoming a member, you'll see how the ICPS is funding different conservation projects around the world. Every other year, we have a in-person conference. 2023 was Japan, 2024 was Vienna, Austria. The reason why those 2 were back-to-back is because Japan's conference got delayed by 4 years, I think, maybe even 5 years because of COVID. And every time we have a conference, it rotates. So we do know that the conference is going to be in the Americas. So that means it could be in North America, but it also can be in Central America or South America in 2026. Right now, the recording, we're doing this in October of 2024, and we should be making an announcement in the spring, late spring of 2025, right? And yeah, I'll leave it at that. So the benefit of going to a conference is you get to go and have lunch and dinner with wonderful people and talk about carnivorous plants for 12 hours at a time. All right, do you agree, David?
Yeah, I absolutely agree. That was a great conference. You may remember similar slides. It was kind of fun because similar slides were being shown at that conference last year, but it was like a translator, right? So every time I said a sentence, then the translator would translate it, and it was fun because, I mean, I don't know. I think the translators were like unanimously like extremely confused as to like the subject. There were so many different subjects, and some people were using like a lot of scientific terms and stuff that were interesting. So, but it was great. It's super accommodating though, even with the language barrier.
Yeah, so David's talking about the Japanese conference, and then the more recent one, the Vienna conference. All of the presentations were in English except for like the opening one, which was in German because it was held by the German speaking Carnivorous Plant Society. All right, so David, I'm going to read some of these questions just because people watching this on YouTube will not be able to see it. And I
Yeah,
do see that
no
you've
problem.
been answering them. Mr. Smith, which could or could not be a real name, says it's not a tongue on the Darlingtonia, it's a mustache. Its actual purpose is to look distinguished. And David, you may use that in future presentations.
I think there is a Photoshop that I made with like a little monocle from a while back. I should have found that.
Brendan says, do you find any commensal insects in the pictures, like these little fly larvae? Now, before you answer that, in November, we have like back-to-back webinars on different collaborators from all over the world. And it's like all about commensalism. All right, so go ahead, David.
Do we find, sorry, what was the question? Do we find any
Commensal insects in the pictures?
in the pictures, like tachinid fly larvae? Yes. So probably. I find them not like as regularly as the midges and mites, but maybe in like 10 to 20%, I often find fly larvae, which I think is another species that is known to only be found in Darlingtonia. And then there's a few moths whose caterpillars are regularly found in the pictures as well. And there's probably other insects that I'm just unaware of.
Okay, Mark adds that the two findings of them in Washington were both in the last couple of years.
Yeah. I thought about trying to go up and visit them. But people I know, like my eyes on the ground in Washington, have already seen them, and they have already told me that they are almost certainly, because there's only like one or two of these plants, and they're not really in any sort of habitat that seems natural. And in fact, they're often in like very populated areas, I think. So yeah, it's suspect. And there's also some records of them in like outside of Victoria, British Columbia, which again planted on like some sort of sphagnum boardwalk area. But I think they're all dead by now, is what I've heard.
All right, so we do have to be careful with what people do. And the next question, we don't want to poach them, but we also don't want to be planting them in their non-native region. Aiden says, are any of your sites facing more poaching pressures than others?
Yeah, I've looked for evidence of poaching, and the only time I've ever found it was probably at Butterfly Valley in Quincy, around Quincy. I spent a lot more time there. And from that poaching, though, it's still quite rare to see from my perspective. And the things that tend to get poached from these sites is more Drosera than Darlingtonia. And I think that might just have to do with the fact that, like, most people probably think it's more trouble than it's worth to be growing Darlingtonia. So that's my explanation for why.
All right, good segue, David. Aiden then says, how do they grow in dry soils? Example, that old gold mine.
Yeah, so again, there was still like a little bit of a trickle of water at that site, which really surprised me. So there's always going to be water moving that is present in some form. Sometimes it's very hard to find, like often it's underground. And I would say that like the sites, so there's a sort of famous site up in Oregon called Bastendorf Bog, which historically had a quite large population in this like clearing with a bunch of rhododendron or azalea. I don't know what it was, but that site has subsequently dried out, and now it's like been totally grown over, and there's almost no Darlingtonia to be found. So I think, yeah, the changes in groundwater flow are really, really important, and understanding how climate change is going to affect them is something that we need to think more about.
And then there's a whole study of fire ecology. And Tim says, knowing many Sarracenia are fire dependent, could we correctly assume that Darlingtonia has some sort of relationship with fire?
Yeah, and so the kind of classic studies have been done by Eric Yules from Humboldt State University on Darlingtonia responses to forest fire up in Humboldt County. Great studies, so I recommend checking those out. But yeah, I think it's probably similar mechanisms. You know, they can first they're in this wet area that probably just doesn't get damaged as much. They are extremely like waxy leaves, and when you throw them in a fire, because you know I spent a lot of time doing that, they don't really burn. They take a long time to burn, and so some of the fire ecologists that I'd be hanging out with at the UC forestry camp where I used to stay, they would be shocked at like how difficult it was to actually burn one of these plants.
All right, BJ says, did you separate out the mites and midges before sequencing, or was it more of a eDNA approach? And then David, what is that environmental DNA? What does that mean?
Yeah, so eDNA is a method where you would just suck up everything out of a pitcher plant and use different genetic barcodes to sequence and identify what species are present. So for the midges, because they're easy to separate out, you can just use tweezers. Every day at the end of the day, I would, throw on an episode of The Wire, sit there with a little microscope and just like sort mites and midges for hours into the night. And yeah, the mites themselves, we got a lot of other stuff. So as we're finding out, as Nono can attest, there's a lot of other DNA in there that we can hopefully use to answer some questions about just the general geographic variation in prey capture and what's found in these things.
So tweezer seems like a real scientist, but when you're putting ants in different liquids, do you feel like a child?
Yeah, it's kind of sadistic. Like I won't lie. I mean, it's I didn't have to get like IACUC approval to be throwing ants into pitcher fluid. But actually, it's really funny because one day, so I was doing these experiments in the lab, and I needed a bunch of ants, and so I ordered online ants from like a reptile supply company or something to feed lizards with. And so this big shipment of ants came, but like over the weekend they had escaped and they had like drilled a hole in the side of the container and like spread all over the mailroom. And so when I went to pick them up, there was a big red sign on the door that said like danger, ants have escaped. And when I went in there were ants everywhere. And so the woman in the mailroom was pissed at me. And I have a photo of it. I should have put that on there too.
Kai says, for the population growing on sphagnum moss, did you find serpentine rock or spring water near it? Was it purely a bog environment?
Yeah, I don't actually know about the geology of coastal Oregon well enough. But yeah, I'd have to see some maps of serpentine outcrops in that area. But just overall, I didn't see any of the sort of like the classic signs, like the green or reddish rocks or anything. It was very different looking habitat. So I'm inclined to say no.
Mark says that he found some Darlingtonia on iNaturalist in the northern coastal Washington. And then this goes back to the other question about how they were found 2 years ago. And we just want to remind everyone to be careful of what they tag and post on iNaturalist. And so I see you commented that they were near a botanical garden and they might not be natural.
Yeah. But yeah, I encourage you to visit them and see for yourself. I would like to know more. And there's no shame in tagging something over and over again on iNaturalist. I do it all the time.
Brendan asked, where and when is it thought that Darlingtonia diverged from Helianthera and Saracenia?
Yeah, so I think based on the phylogeny I showed, something like, I don't remember, 50 million years ago, or maybe I'm thinking about the ferns I study. But yeah, I really recommend you read this paper. So Aaron Ellison is the first author, and Chuck Davis from the Harvard Herbarium, and think Rob Noxie is on the paper too. It's in PLOS One, so it's freely open access. I think it was published around 2010, 2011. So check that paper out because they go into the plate tectonic movements and timing of how that might have occurred as well. So yeah, really good paper.
And for those of you who don't know, Aaron Ellison, he did a ICPS webinar 2 years ago, which you can watch for free on youtube.com. All right, and I think the last question of the night is from Mr. Smith. Are there efforts to preserve representatives from each population and cultivation? And if you went to the Vienna conference this year, you would have had a heated debate and argument about this from Andreas Fleischmann. But David, what would you like to say?
I don't think there are any efforts whatsoever. I know that there's efforts to find the easiest to grow cultivars and just clonally reproduce those into oblivion. But yeah, I don't think there's much going on in terms of ex situ cultivation, because again, the general sentiment is that they're hard to grow. They don't really grow outside very easily in most places. And so I think people are just kind of afraid of doing it, to be honest. I don't know. I'm certainly afraid of growing them.
So that was Mr. Smith's question, and then he added that he loves Andreas. And I'm gonna kind of butcher his summary, but Andreas was basically saying there's no need to be saving in cultivation each of these representatives because you cannot save all of those organisms that are living on and in those plants. And if we're killing the environment, there's no place for those plants to go back to.
Yeah. I mean, that's kind of what my maps at the end suggested as well. So definitely we should be conserving the sites more than just the plants themselves.
David, I wrote a question somewhere. When you did those projection maps, was that based off of somebody else's plant work, or is that a novel thing, and are other species or groups doing that?
Yeah, so I basically made those maps just with my own GPS coordinates, and then I did the statistical modeling myself. But I mean, it's a commonly used technique in ecology to forecast species distributions, and so for everything. So I'm doing it with all the plants in Japan right now as well. I'm trying to make a big compendium of projected plant movements in Japan. But it's quite common, yeah.
Thank you everyone for joining us live, and I hope to see you next time.
It's not a surprise that gardeners, educators, and scientists are fascinated by these unique plants. The International Carnivorous Plant Society, or ICPS, not only love these plants, but welcomes growers just getting started all the way through professional scientists. The ICPS even started an annual World Carnivorous Plant Day to celebrate them. The free online event is held the first Wednesday of May. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.